Hi, I'm Janika Ruuska and I'm studying Neuroscience at the University of Helsinki in Finland. Currently, I'm in Australia and I just finished my master's thesis. So in today's episode, I will tell you how did I end up here, how it was to do my thesis, what was the topic and how I studied that, and what were the most useful tools while working with my thesis. Welcome to today's episode. Alright, so let's start from the facts, how I ended up here in Australia to do my thesis. I would say over a year ago, I was doing my internship back in my home university in one research group where we studied the anxiety disorders. I learned a lot about the topic, about the methods, how to study that, but somehow I still wondered, is this the topic I'm the most interested in? And would there be something else which would be closer to my passion? Back then, I had a discussion with my supervisor. We talked about, for example, the topics I'm interested in. And one day, she found one research group from Twitter. And in fact, it was the research group I finally ended up to do my thesis. So thanks for me doing self-reflection, following my passion, talking about that openly, and obviously the help what I got from other people to find these places and giving me a chance to follow my passion. I need to mention that even though now it might sound easy thing to do, but it wasn't. It was a lot of work, but I would still do that all over again. Obviously, I need to apply the position and have the interviews, which are the basic things. But since this country has certain requirements, such as visa, I needed to apply that while we still had some sort of travel restrictions. So it wasn't an easy thing to do, and I waited a long time. And it's not like, it takes time to fill all your information and find all the documents which support your visa application. So only that alone was quite a lot of work. Then obviously I needed to move all my stuff, pack everything, travel here, and then start to figure out all the basic things like mobile data, bank account, public transportation, apartment, and so on. So quite a lot of these practical things I needed to do to be able to conduct my thesis. So this whole process wasn't organized for me. I organized it from the beginning till the end. But as I said, I would do it all over again, and if someone else is gonna do that, I'm happy to share all the tips. Alright, next I wanna tell you how the life was while I did the thesis. To be realistic, to not romanticize the whole image, I needed to learn 3 new topics. Not just 1, 3. I will go a bit more in depth to these topics in a bit, but basically I needed to understand about eating disorders, psychedelics, and methods, how to study these questions from the neuroscientific point of view. So 3 topics, and I had 6 months to do that. And as you can guess, it's not just that you read and try to understand everything. You need to also do the writing, and the writing needed to be like in a publication format, so you needed to learn how to write. Then you need to have all that supporting material like creating illustrations and crafts, and to get any kind of results you need to do the experiments. So learn new methodologies, optimize everything in your protocol, then you will conduct the protocol, and then you start analyzing everything, and then you try to understand everything. And all this happened in 6 months. So it wasn't an easy job to do. So for me personally, it was really, really important that I was really passionate about the topic, because the hours I worked with this were insane. But because I had this craving to learn more, I actually had the energy to do that. And whoever does any kind of thesis may recognize this feeling that if you have a tiny bit more relaxing period when you are working, you might get these thoughts that you are not doing enough. Maybe even you are thinking that you're lazy. I got those. And I got those even I knew that this relaxing time, it wasn't actually relaxing, but more relaxing time, will not last forever. I still had those thoughts, even though I knew it. And it's always funny, because then the really, really intense period starts, and then you are like, yeah, I knew this will happen. Why I didn't enjoy the time? Okay, this was a little bit sidetrack story. So let's go back to how the life was while I did my thesis. In addition to these practical things and doing the experiments and writing and reading, I spent quite a lot of time to communicate with people, to organize things, and to solve problems. Because things will not go smoothly, ever. So if I do a little bit self-reflection, what all I learned while I did my thesis, I learned how important it is to have clear communication and kind of communicating more than even needed, especially if you are doing any kind of collaboration. Because every people have their own things in work life and personal life. So what I mean with this is, even though someone could be able to think, okay, I should do this because I know that the next person will do this, and that might happen, but it's always good to have some kind of clear structure, what will happen, and communicate about that. And also about what I have already done, so people are aware where we are in with that project. And so I'm really organized person, and I'm looking the bigger picture, looking a bit more forward than where we are now, being ready for the problems what might appear. The thing what I still needed to learn, even though I have worked in science for years, so I know that things will not go smoothly, there will be problems. I still needed to remind myself that I need to accept that this will happen. There will be delays, but in the end, things will be solved. One even more important thing what I needed to remind myself was that I'm human being. I'm not a robot. So yeah, I think I need to keep reminding myself about that. And what I mean by that is that I'm learning. Every one of us are learning. And that's part of the beauty, especially in science, that there is always something we can learn, become better. We will not be ready ever. And that's actually the thing which excites me in science. And I bet that most of the people in science have the similar mindset, that they enjoy this. Still, quite many have the thoughts like self-doubting thought, or I should be better in this, kind of a little bit harsh to ourselves. So maybe I will say this to everyone, that we are human beings and we are learning, and that's okay. So more important than being perfect from the beginning is that you're willing to learn, the attitude what you have. Because I had so short time to do all this, I kind of accepted that I will not have too much social life, I will not have time to enjoy other things in life. I still want to remind how important it is to have even like super small breaks. So obviously I don't encourage people to have that hectic and stressful work-focused life, that there is no space for you. But sometimes, if you know it will last only short time, it might be okay to push yourself through that time, work really hard. We all know ourselves best, so I knew I'm able to do that. But still, there were times when I worked many weeks without any break. And then I realized that, okay, even though I would sleep enough, I'm exhausted. So just having a little bit, few hours kind of free time to do something you enjoy where you can't think your work was really important and gave me energy to continue on the next day. So if you have a short time, you need to work really hard. There's a lot of pressure. You might know you can do that, but still have a tiny bit free time. One thing what I think it's interesting to pay attention while I was doing my thesis was to understand what are the skills I still need to practice a little bit more, because I have some strengths and some things still needs to be practiced more. So kind of pay attention to those, so when you take the next step you can focus for more those things and become better on those. But also pay attention what are your strengths and if you want to carry them to your next steps and what you are interested in about. Keeping this in back of your head while you are doing things, it's really important, because then when something ends and you need to think what next, you can do some self-reflection and maybe that guides you forward. So basically I try to summarize for you what I studied. So my topic, as I mentioned, the main topic was actually feeding and eating disorders and psychedelics and how to study that, but a little bit more detailed. So I focused to anorexia nervosa. And when we talk about disordered eating, it means that the person has continuous and disturbed eating behavior, which leads to impairments of physical health and or psychosocial function. So basically in anorexia nervosa, this means that the person is eating less than their body needs, which often leads to losing weight. And this is really serious condition, and there is a lot of rigid thinking about eating and exercising, about body. It's really complex, and I'm not able to describe completely now, so I think I will do another episode about that. But that was one of my topics. As you can guess, because it's so complex disorder, that there are many things you can study and try to understand better. So mine was focusing to the molecular level of this disorder, and there are still many options. But about the second topic, so I studied psychedelics, and actually I studied psilocybin, which is found from more than 100 species of mushrooms, and its active metabolite psilocin is known to alter, for example, serotonin function. So it has high affinity towards several serotonin receptors. So there are many other things and other receptors it's known to cause the effect, but I focused to serotonin receptors, few subtypes of those. And I was mainly interested in about prefrontal cortex, because we also know that in anorexia, the activity of prefrontal cortex is heightened, so kind of more active, and possibly because of the altered serotonin activity. So the idea was that maybe we could affect that by using psilocybin. The method what I used to study these questions, so investigating the effect of psilocybin in anorexia nervosa, I focused to several serotonin receptors, as I mentioned, and I used a method called Erenescope, which is novel in situ hybridization method, with what we can study the gene expression for the receptors of interests. So really tiny mRNA molecules can be studied by using this method called Erenescope. And with that we can show the localization and the expression in the certain brain area. Well, if I give a more general idea about the Erenescope, it's basically you do this fairly long protocol Basically, it took 10 hours in one day just to process few samples. But anyway, the results are super cool. So you have several probes, which tells you about different gene expressions. Then you use the fluorescent labels, and those will send a different kind of color. In a microscope you see it in different color, so it depends from the light wavelength. So you will see them as a different color. The different receptors will be different color. This is the easiest way to say it. So you could have, for example, blue, and then you can have a red, and then you have a green, and then you see those different dots representing the mRNA molecules what you are studying. And to analyze the results, we used Cell Profiler and a pipeline there. By the way, you will see all these things better explained in my thesis when it's out. So if you are more interested in what kind of pipelines or all that we used, you can see them from there. But basically this pipeline was really useful because I didn't need to count manually how many dots are in one sample. It would be crazy amount of work, and also there is risks for error because there were so many dots, and if I have any kind of expectation, that can also affect to my counting. So when we automate this, it's more, you can trust the results more. So the pipeline, there are several public pipelines you can use, and you can modify them, and you just need to know what are your research questions, and you can kind of tell to the program with the pipeline that these questions I would need to be answered. For example, how many dots are in this sample? So to summarize everything, anorexia nervosa, prefrontal cortex, few serotonin receptors, and how psilocybin affected those. Finally, I want to share the tools I found the most useful while I was doing all this, from the experiments to the writing to the reading and beyond. So I gotta mention Word. So just to creating your documents, this is probably pretty obvious. So I did writing by using Word, but I also use it for creating protocols. And the reason why I want to mention this is that sometimes we might use protocols from some research paper, or we have learned them by someone showing that to us. So I would highly recommend to have the protocol in a protocol form. And if you have protocol from someone else and you have done any modifications, just create a new one and put the notes that you have edited it from this certain protocol. And I had all the protocols in one folder, and that made it so much easier for everyone who wants to see them later on, that how I have done things. Yeah, obviously I had also the notes in lab archives, so I had a physical notebook, and then I had electronic version, so they will not disappear anywhere. So these are really basic tools, and also Excel that I used a lot. I used it from the beginning when I read the papers. I have talked about this previously, so I recommend to watch that episode. But so I collected all the papers I read, and I marked which papers I actually used in my thesis, and I had some notes there, what were the main things this paper talked about. So it was easier to go back if I needed some certain information, kind of find it from my Excel sheet that, oh, it's in this paper. But I also created a lot of different Excel sheets, so just basically collecting all my data for there, but also the information about my samples, if there has been any mistakes, anything weird happening, just to write those notes there, where you can find them, how they are labeled. I also had a different sheet for reagents. I put the reference numbers and from which company those are, so if they are running out, we can easily find it from this certain Excel sheet and order more if we need it. And actually, what comes to the applications, I started to use Mendeley, if I remember the name correctly, in the beginning. So kind of collecting all the papers in one place. You can do some notes there, highlight things, and you can use, if you use Word for writing, you can have the plugin there to the Mendeley plugin. So it creates the reference list in that style you want. But somehow I had some issues with that. I can't even remember, but I got a bit annoyed, probably because it was locking me out or something like that. So I switched to Zotero later on, and I have used that already previously. So I shift back to that and it's working. So I would maybe recommend that instead of the Mendeley. What comes to creating graphs, I used GraphPad with what you can also do some of the statistics. And that's quite easy to use. It took a little bit time to understand how it works, but when you have done the work once, then it's really easy to use, and it doesn't allow you to do kind of wrong type of statistics from your tables, what you're creating there. So I use that, and you can change the colors so nice. I love how things looks. So it was also really easy. They have these presets for things and fonts and all that. I created all my images by using Render. It's actually, I think it's quite new program. It hasn't been too long. Well, it's not new, but like fairly new. But it's really nice. I guess most of the universities has the access to the paid version, and they are kind of these ready-made images, for example in my case about the prefrontal cortex, and then there are some like basic lab equipment. I want to mention that for communicating with people I used email. I'm a bit old-fashioned, but that was basically for things like longer messages and kind of confirming things and ordering things. But then I used Slack for communicating some quick things, and I found this is really important, kind of, at least for me, to keep my personal life communication somewhere else, and then I have this work-related communication in some channels. I find that's really easy. Then I have things in one place or two places. Yeah, email and Slack. And this is really basic thing, and I bet everyone knows this. This should be done, but still things happens, and maybe people think I will do that later and all that. So save things to cloud, and if possible to hard drive. Save it for several clouds. So when you are writing your thesis, save it to everywhere, and save it to save million versions of the things you are creating. Create a copy of your thesis to cloud, and then next day you're working with that, create another copy and continue from there. So you have these many versions from many different locations, just in case if something goes wrong. Luckily this didn't happen in my case, so I had it everywhere, but it somehow gave me a peaceful mind when I knew that there is no risk it will disappear. When thinking about the devices, it of course depends if there is possibility to have certain devices, but if possible, I really enjoyed, I had my MacBook with what I worked most of the time, and I have always thought that when going further in a scientific career, if possible, get as good computer as possible with good power, because we need a lot of programs which will reserve a lot of memory and the capacity of the computer, and it's so much better when the computer is fast. So I was, for example, running a VG, ImageJ, with the plugins, and then I was using the cell profiler, and yeah, it was a heavy program, or heavy pipeline, I would say, with the heavy images, from the microscope. So I was doing that at the same time when I was doing some writing or reading. So if it would be lacking all the time, it would be super annoying. Anyways, extra tip, whatever computer you have, you can use. If you have possibility to have another screen, have that. That was really, really useful. Kind of have your things spread there, and then you can focus the thing what you are actually doing. Maybe have your notes on the other screen, and you don't need to like do any other extra movements. Just like turn your head and look what is there. And I used iPad for the first time. I found it really useful. I liked the fact that I was able to read papers on my way work. I could do some notes. And also when I was creating some of the images, somehow I enjoyed to work with iPad because I did some like small coloring details with the pen. So to summarize the tools, what I used. So Word, Excel, basic like cloud or hard drive, save million times. If you have extra screen for your computer, that's perfect. Have a Slack or use email. Maybe Zotero for publication collections. BioRender for creating images. And GraphPad could be a good option for statistics and graphs. So yeah, this is how I survived through my thesis. And yeah, I would recommend, if possible, whoever is free in the work group you are working, or maybe your friends, let them read through your thesis and get all the comments possible, what you should fix, or if something is not understandable, and accept that you will have all kind of emotions and trust that you will go through it, and try to be excited about small things, small achievements. Since now I have submitted my thesis, it means that it's not too long. I will graduate and then I'm neuroscientist instead of future neuroscientist. So there will be few episodes because I don't want to leave you yet, but that's gonna happen, or then I will create something new. But I'm super happy that you have been here joining to this journey with me. Hope you will still enjoy the future episodes. So see you on next time. Bye.